Hi everyone, in this video I'd like to talk about the structure of nucleotides and the structure of DNA. And we're going to be emphasizing throughout two things. One is 4 different points about the structure, and then finally 4 rules of DNA. The way we're going to do this is we're going to start out by talking about a children's toy, and then we're going to move on to making kind of a cartoon simplified model, and then we're going to move on finally to the actual chemical nucleotides. So the point throughout is to get a sense of the structure with something very concrete, so that we can take this thing that seems very abstract and think about it in a very concrete way. All right, so let's jump right in. So here's the toy we're going to start with. Some of you may have seen this. It's a happy birthday sign. And what you can see is that it's made up of different letters of different colors. If we zoom in on those letters, what we see is that each one, that it's a chain of kind of similar things where we have a letter, and each letter has 2 sides. It has a hole, and then it has a peg. And everything is joined together by the hole goes in the peg, the hole goes in the peg. And here it is in close-up where you can see one junction. Okay, so let's make a toy model of this so that we can talk about it a little bit more. And so a couple of points that I want to make about this that also apply to DNA and nucleotides is that first, this chain of letters has 2 different components, right? 2 different parts. Down here it has the informational part where we've got the different letters that we can actually use to spell words. But it also has this structural part, the kind of the boring part that just is necessary to link everything together. And in fact, we can talk about the structural part as being like a backbone. This is the metaphor we use for DNA. We talk about the DNA backbone, which is the structural part that holds the nucleotides together. And so it kind of makes sense to think about a backbone. The vertebrae in a backbone are all very similar and come together very similarly. And you can see their structural similarities. But then each one has its own independent features. So in the same way that each letter here has the backbone part that hooks together, it also has these appendages off of it, the H, the A, and the P, which have these specific functions. The next point I'd like to make that also applies to nucleotides is that each letter has 2 sides. It has a loop and it has a hook. And the hook goes with the loop and the hook goes with the loop to make a chain. A hook does not go with a hook and a loop does not go with a loop. Okay? The third thing is that because of this hook-loop structure, all members of the chain are in the same direction. So if the H goes from loop to hook, then the A goes from loop to hook, and the P goes from loop to hook. So those are the 3 points I want to make about that for right now, and let's look at some other toys that can kind of help us to gather an intuition for this. So those of you who are familiar with Legos, this will make sense. If you're not, basically Legos, you can see they have 2 ends. One end has pegs and the other end has holes, or over here pegs and then on the bottom holes. And when we play with them, we stick them together and the pegs of one fit into the holes of the one above it and so forth. So we can stack everything up, and if we looked at it in X-ray vision, we would see that it goes pegs into holes, pegs into holes, pegs into holes, pegs into holes. Right? So this is another thing where we have a structure that has 2 ends and they're all facing in the same direction. Pegs don't go with pegs and holes don't go with holes. Another toy is actually very similar to Legos. These are pop blocks. You can see they have a hole here and then they have a peg, and again here's the hole and the peg sticks into the hole, peg sticks into the hole to make a chain. Another thing we could think about would be like a conga line, right? If everyone's standing in a line together, holding onto the person in front of them, they're all standing in the same direction. Everybody's front is facing the same direction, everybody's back is facing the same direction, all the same direction, not this green person in the opposite direction. And another would be like stacking chairs. When you stack chairs, of course, you stack the bottom of one on the top of the other, not the bottom on the bottom or the top on the top. Okay, so let's move from this toy into an actual model of a nucleotide. So our hook and loop we can make a structure out of it. And we can change our H to one of the 4 nucleotides, A, C, G, and T. And so then we would have our 4 nucleotides. Notice that they have different informational components, but that their backbone parts are identical. Because these are actual chemicals, it makes sense to draw them in different structures. You can see that the 4 have different structures. And we'll get back to sort of how that works out. So again, we can see that we have the structural part, the backbone, and the informational part that actually carries the difference between nucleotides. Again, we have 2 sides. We have a 5' and a 3'. And we can see each nucleotide has 5' and 3'. And then these things fit together with the 5 going with the 3, the 5 going with the 3. The 3 does not go with the 3. And the 5 does not go with the 5. And so what this means is that when we put them together, we can see all of our nucleotides go in the same direction, 5 to 3, 5 to 3, 5 to 3, and 5 to 3. Okay, so let's talk quickly about a rule for writing nucleotides. Why don't we all agree that when we write things, we're going to say that what we mean is the 5' end to the left and the 3' end to the right. So if we just write A, that means an A with its 5' facing to the left and its 3' facing to the right. So if we need to write an A with a 3' to the left, then we can write it upside down. So let's look at what that means then. That means if we write A, we mean an A oriented this way. And if we want to write it in the opposite direction with the 5' to the right, then we can just write it upside down. Okay, so whenever you write an A, you mean the 5' is before that and the 3' is after it. So if we write it upside down, then that would mean the 5' is over here and the 3' is over here. And so if we want to write out a nucleotide base pair of T pairing with A, then that would mean that what we do is we write the T, which goes 5' to 3. So we write it right side up. And we're going to write the A, which is upside down, which has the 5' to the right. We're going to write that upside down. And so if we wanted to write a short sequence of CAGT on one strand or ACTG on the other strand, we can write it like this. Okay, so if you write CAG, I'm going to assume that what you mean is 5' CAG 3'. And so we have our final point here, which did not apply in the happy birthday case, which is that nucleotides on pairing strands are in opposite orientations, right? So now we have a double-stranded, this is our first model of a double-stranded piece of DNA, and we can see on the top, all the nucleotides are 5' to 3', and on the bottom, they're all going in the opposite direction with the 5' at the right and the 3' at the left. Okay, so now we can move on into an actual DNA structure. So now we have 3 different nucleotides. And so again, let's go through our points to make. So our first point is that we have 2 parts, and we can break this apart. If we break it apart, we see that the blue, which is one nucleotide, that it has an identical structure to the orange one and to the black one, and the differences are just in this region. And so what that means is that we've got the structural part, the sort of boring, homogeneous repeating part. And then we have the informational part, which are these bases that stick off of the base sugar. So then the second point to make is that each nucleotide has 2 sides. So we said this is 5' and 3'. So if we zoom in on one, we can try to remember, as you might have learned in chemistry, why these are called 5' and 3'. And so here's the base, here's the informational part. And so because the base is the biggest side chain, the biggest molecule here, we're going to number that carbon 1', and then the next one 2', 3', 4', and then this one is 5'. Okay, so this is what we mean by 5', and this is what we mean by 3'. And so then if we focus in on our nucleotides, what we can see is that the bonds are between the side chain off the 5' with the oxygen off the 3', or sorry, just the just the bond to the 3'. And then the 5' of this nucleotide is bonding to the 3' of that nucleotide, and on and on. Okay, so we have the two sides, and we have this this situation where the hook binds to the loop, and the hook binds to the loop. The 5' binds to the 3'. The 5' binds to the 3'. Not 5' with 5', not 3' with 3'. And so what that means is, once again, our third point, is that everything is in the same, everything in a chain is in the same orientation. This nucleotide is 5' to 3', this nucleotide is 5' to 3', this nucleotide is 5' to 3'. And so now I've just rotated this to the left. I've flipped it up, pardon me, I've flipped it up like this. And so now we can see how this relates to our initial model. So here we have thymine, guanine, cytosine, and adenine. And so now we can do our fourth point, which is that nucleotides on pairing strands are in opposite directions, by adding in our second strand. So here we have the red, let me remember, the red thymine here is pairing with the orange adenine, T with A. Here we have G with C, and then C with G, and then A with T. And we can see that the top strand is going 5' to 3', and the bottom strand is going in the opposite direction, from right to left, 5' to 3'. So here's an exercise that I suggest you do. Make some nucleotides out of paper. So that would mean just have a box of paper that says 5', A, 3', and then another one with C, another one with G, another one with T. And actually for this exercise, probably one of which will be different. And then make the sequences 5' ATG3 and 3' ATG5, and see, just have a look at them and see that they're not the same structure, they're not the same sequence, they're a different relationship between the nucleotides. So I'll leave that for you to do and just continue on with the video. So now I want to move into the 4 rules. So some of these you've already seen while we were making the points. But if you really can learn these 4 rules well, you're going to know nucleotide structure better than you did before. So the first thing is, in a single strand of DNA, all nucleotides are in the same direction. Double strands are in opposite directions. And then you probably have seen before, and we touched on it briefly, complementarity. So an A will always pair with a T with an A C with a G, and a G with a C. And so this 4th rule is new. And this 4th rule has to do not with what's the structure of DNA, but how is DNA and RNA built. So here we have on the bottom a long strand of DNA, and on the top a strand of DNA that is newly forming. So you can see it's just these 4 nucleotides. And our question then would be, when a new nucleotide comes in, where is it going to bind? Is it going to bind over here, or is it going to bind over here? And this is where this rule comes in really handy. And so the rule is that nucleotides are always added to the 3' end of the strand. So this nucleotide is going to come and bind over here, and we are not going to add nucleotides to the 5'. All right, so the new nucleotide comes in, and now the next nucleotide is going to bind over here, not over here, because this is still the 3' end, and then the next nucleotide would get added here. And so if we think about what that looks like, that means we started with the T, then we added the G, then we added the C, then we added the A, then we added the A, then we added the A. So this is what you've heard of as being 5' to 3' growth. But I really strongly suggest thinking about it in terms of that nucleotides are added to the 3' end. And the reason for that is, if we look at this double-stranded DNA, what we see is that the strand that's growing is going 5' to 3', but relative to the opposite strand, it's moving from that strand's 3' towards its 5'. And students get very confused with this. So I came up with this rule as a way of making this straight. So instead of saying 5' to 3', I strongly suggest that you say that molecules grow on their 3' ends. If you can learn it that way, I think you'll be in much better shape. Okay, so just summing up, we talked about these 2 2s. We had our 4 points, but 2 of them overlap with the 4 rules. So let's just talk about the 2 2s. Nucleotides have 2 parts, an informational part and a structural part, and they have 2 ends, a 5' end and a 3' end. Then we have our 4 rules. Nucleotides in the same strand or in the same direction. So here's a nucleotide that's 5' to 3', 5' to 3', all in the same direction. Nucleotides in pairing strands are in opposite directions. We have complementarity, where A and T pair with each other, and G and C pair with each other. And finally, perhaps the most unfamiliar one, is that nucleotides are only added to the 3' end. Thanks very much.